Good morning. Welcome to the Eurofins Discovery webinar for the Japanese Society of Toxicology. My name is David Wei. I am currently the Director of Drug Discovery Partnership, and I will be serving as your moderator today. Today, we will be hearing a presentation from Dr. Panida Lakyamohon, who will be speaking to us on validation of the high-throughput assay using human iPSC-derived cardiomyocytes to assess cardiotoxic compounds under the CiPA initiative. Panida is currently a senior scientist and group leader of cell-based screening team of Eurofins Discovery, where her main responsibility is to manage cell-based screens for in vitro safety pharmacology profiling panels and high-throughput screening campaigns. She works with clients that range from small biotech to large pharma. Prior to joining Eurofins, she was a postdoc at Stanford Brigham Prebys Medical Discovery Institute, performing cell-based and biochemical assays for drug discovery and enzyme kinetic analysis. With over a decade of experience, Panida's expertise includes high-throughput screening, mammalian tissue culture, protein ligand bindings, and other in vitro assays. Now please welcome Panida for her presentation.
Thank you, David, for the kind introduction of myself. Good morning, everyone. My name is Panida Lakyamohon. I'm a senior scientist and a group leader of a cell-based team from Eurofins Discovery at San Diego, California, USA. I will be presenting an application of human iPSC-derived cardiomyocytes to assess cardiotoxicity of compounds. This paradigm is engaged by comprehensive in vitro proarrhythmia assay, or CiPA, for cardiac safety during drug development. Our agenda for today will include background on cardiac and ion channels, how ERG is important for cardiac safety, and how it has become a limitation in drug discovery. Comprehensive in vitro proarrhythmia, or CiPA, is a new paradigm for cardiac safety evaluation and how Eurofins Discovery takes part in CiPA initiative. I will discuss about our calcium flux assay service using human iPSC-derived cardiomyocytes that I will show our validation data. Then, we will conclude the talk with the application of cardiomyocytes in cardiotoxicity assessment and Eurofins Discovery CiPA initiative, CiPA assay services. Let's get to know our hearts. The heart is a pump that is driven by electricity. In a minute, it can beat from 60 to 80 times. That means that it's about 100,000 times in a day, or 35 million times a year, or 3 billion times in a lifetime. Therefore, if beating of the heart is interrupted more than just a few minutes, that is detrimental as multiple organs can be damaged. It can even cost us alive. We can look at activity of the heart by measuring electrical activity of the heartbeat. This starts with an electrical impulse that travels from the upper chambers of the heart to the lower chambers. This electrical impulse stimulates the heart muscles to pump blood from the heart. The electrical impulses are initiated by the upper chambers that generate the first wave called P wave. After that, the bottom chambers generate the next wave, namely QRS complex. The final T wave represents the electrical recovery, where the bottom chambers return to the resting state. Consequently, if these waves are distorted, function of the heart can be abnormal. Ion channels expressed in the heart are the key to the healthy waves. The heart expresses several ion channels. ERG, or human ether agogo-related gene, is a potassium channel that relates relaxation signal or repolarization to the heart muscles. ERG is promiscuous as it can bind to diverse kinds of compounds. Inhibition of ERG can prolong the QT interval. However, this is not detrimental unless early after depolarization or EAD occurs. Opening of calcium channel or late sodium current during the prolonged phase can result in EAD, leading to Torsades de Pointes or TDP. Therefore, ERK inhibition can lead to QT prolongation, but only together with EAD, it can lead to TDP. Drug-induced QT interval prolongation and Torsades de Pointes arrhythmias are the leading causes for compound attrition during drug development. In 1998, tafenidine, which is a non-sedating antihistamine that can induce QT prolongation and arrhythmia. In addition, sertindole is also another drug which is an antipsychotic drug that can induce QT prolongation. Then, in 1999, acetaminophen, which is a non-sedating antihistamine, was found to induce QT prolongation and arrhythmia. And there are many other drugs that are found to induce QT prolongation, such as in 2000, cisaprine, which is a gastroprokinetic agent, was found to induce QT prolongation and arrhythmia. Therefore, in the year of 2000, S7B was introduced as a guideline for non-clinical testing strategy to assess the potential of compounds to delay ventricular repolarization. These example drugs are pro-ERK blockers and highly selective in the therapeutic ranges, since the events of TDP are far too low to be detected in clinical trials. QT prolongation is used as a surrogate marker. Then, how can we relate IC50 of ERK to cardiac safety in clinical trials? This is the quote that poison is in everything, and nothing is without poison. The dosage makes it either a poison or a remedy. We use safety margin, which is calculated from IC50 divided by free plasma concentration. For ERK, if the safety margin of a compound is over 100, then the compound is preferred for further development. But if the ratio is below 30, the compound shouldn't be continued for further development. Eurofins discovery offers an assay to estimate free plasma concentration of compounds together with ERK inhibition assay for evaluation of safety margin. Since blocking ERK nor QT prolongation does not necessarily mean TDP, comprehensive in vitro proarrhythmia assay, or CiPA, is an initiative to facilitate adoption of a new paradigm to assess clinical potential of TDP. The new CiPA paradigm is comprised of mechanistically based in vitro assays coupled to in silico reconstructions of cellular cardiac electrophysiologic activity. Then, verified completeness through comparison of predicted and observed responses in human-derived cardiomyocytes. In correspondence to the need for proarrhythmia assessment, Eurofins Discovery participated in the HESI Cardiac Safety Proarrhythmia FDA high-throughput subteam in 2015 to assess variability and reproducibility of high-throughput screening platforms or sites for defining drug effects on cardiac ionic currents across and between sites in an effort to support CiPA initiative. There are 28 compounds categorized as no or very low risk for TDP, intermediate risk, and high risk for TDP. These test compounds are blinded and shipped from one source. Then standardized assay protocols were employed to collect data from different platforms. To further verify clinical potential TDP of compounds, human-induced pluripotent stem cell-derived cardiomyocytes are used to mimic effects of the compounds against heart muscles. To obtain iPSC-derived cardiomyocytes, skin samples are collected and grown in cultures. Then, 4 genes were introduced into the skin cells, and these cells differentiate into cardiomyocytes. iPSC-derived cardiomyocytes can be used as a model to evaluate potential cardiotoxicity of compounds. Using cardiomyocytes as a model, we can perform calcium flux assay to monitor contraction and relaxation of cardiomyocytes. We detect either increased or decreased levels of calcium ions using calcium-sensitive dye. The dye binds calcium ions in cytoplasm as displayed on the left panel. The dye elicits fluorescent signals that can be captured by a high-speed CCD camera in FLIPR, which is a real-time imaging platform. Shown on the left panel is the picture of iPSC-derived cardiomyocytes from Fujifilm CDI. I grow these cells in a 384 well plate for the calcium flux assays and used the new FLIPR Penta, which is equipped with a high-speed camera that can capture 30 frames per second, compared to the previous Tetra that can go up to 6 frames per second. This allows us to obtain high-resolution data as displayed on the left. The top panel is the vehicle control, and the lower panel is the xylitol-treated cells. You can see that xylitol, which is a high-risk HTP compound, causes prolongation of the calcium oscillations compared to the vehicle control. To define calcium oscillation traces, we choose 3 descriptors, which are mean peak rate that demonstrates how many primary peaks observed in a minute. Peak spacing is the space from the beginning until the end of the peak, and calcium transient duration 90 or CTD 90. Moreover, we can also observe the secondary calcium spike that is an EAD-like waveform. The detected fluorescent signals from the calcium flux assay represent intracellular calcium oscillations. In order to understand what happens to cardiomyocytes upon compound treatment, I use PeakPro2 displayed on the left panel. PeakPro2 is a model of ScreenWorks software of FLIPR that we can use for extracting kinetic parameters from the calcium traces. PeakPro2 can assign primary peaks, which are identified by blue dots. It can also set the baseline and the triggered levels, as shown in red and green lines. The increase in fluorescent signals corresponds to contraction, while decrease corresponds to relaxation of cardiomyocytes. We can monitor in real time and over time. In this example, the second panel on the left is the pre-treatment trace. Then the cells were treated with compounds and incubated for 30 minutes and read on FLIPR as shown in the third panel on the left. There are several parameters that we can extract from the traces using PeakPro2. From our experience, mean peak rate, CTD 90, and peak spacing are pretty consistent across the whole 384 well plate and from experiment to experiment. So we can go ahead with these parameters to plot our dose-response curves for calculating RC50s as shown in the right column. There are more examples of cardiomyocytes treated with isoproterenol, which is an agonist of beta-adrenergic receptor that can increase the heart rate. The trace exhibits increased mean peak rate and decreased CTD 90 and peak spacing. The next one is astemazole, a non-sedating antihistamine that is categorized as intermediate risk of TDP. Astemazole is a point ERG blocker and causes QT prolongation from the trace. The mean peak rate is decreased, while CTD90 and peak spacing are increased, which are characteristics of peak prolongation due to blockage of repolarization by ERG. The last one is xylitol, which is categorized as high risk for TDP. Xylitol is a beta blocker. It can also prolong repolarization by blocking the rapid component of delayed rectifier potassium current. From our assay, xylitol-treated cells exhibit decreased mean peak rate but increased CTD90 and peak spacing, showing peak prolongation. For these three parameters, the data points are quite tight and clearly distinguishable between pre-treatment, shown in brown, and post-treatment, shown in navy. These scatter plots demonstrate that our calcium flux assay is uniform and consistent. The observed effects are also correlated with actions of the compounds. Using our assay, we can monitor real-time cardiotoxicity of E4031 at different time points from low to high concentration. At low concentration of 8 nanomolar on the top row, we observe peak prolongation and secondary calcium spikes at 30 minutes post-treatment, while at very high concentration, such as 500 nanomolar, at 30 minutes post-treatment, we observe cardiac arrest. The effect of 500 nanomolar is very fast that we can observe peak prolongation as early as 20 seconds post-treatment before the cells undergoing cardiac arrest. Again, this is to reiterate that we can observe either stimulating or blocking effects of compounds compared to the cells treated with the vehicle control. Moreover, the secondary calcium spikes, which are EAD-like waveforms, are observed in the cells treated with xylitol, which is a high-risk TDP compound. We can demonstrate that treatment with xylitol results in peak prolongation and secondary calcium spikes correlating with its high-risk TDP. We can also plot dose response curves and fit the curves of these three parameters to obtain EC50s or IC50s. As you can see that across different parameters, the resulting EC50s or IC50s are pretty much in the same range. In summary, we can see that the heart can pump blood normally as long as the beating is not distorted. Irregular beating can be detrimental, especially in the event of TDP. Therefore, drug-induced QT prolongation and TDP should be considered to reduce the chance of compound attrition during drug development. And CiPA initiative is a new paradigm for TDP risk assessment. For us, Eurofins Discovery, we provide services to evaluate cardiotoxicity of compounds as engaged by CiPA initiative. We previously shown in the slides that we can use human iPSC-derived cardiomyocytes to assess cardiotoxicity of compounds through our calcium flux assays. In this assay, we can get phenotypic results of and validate the assay, which can be helpful to mitigate TDP risk during drug development. And thank you for your attention. Any questions, please refer to the please refer to the email as shown in this slide.
Thank you very much, Panita, for addressing this very interesting topic. We have a question for you. What will be the main application of this human iPSC cardiomyocytes FLIPR assay in drug discovery?
So there can be two aspects of the assays. First of all, we can use our assays to assess cardiotoxicity of the compounds, whether the compounds would affect the function of heart muscles, regardless of specific ion channels, since the heart muscles, they have all the ion channels expressed. So then we don't have to target any specific ion channel to see if the drug would have the effect, which is a benefit of using cardiomyocytes. And the other thing that we can use this assay for is to screen for phenotypic results that we aim for. So we provide a service that it will be for concentrations in triplicates. So then you can see whether the drug would have the inhibitory effect or the stimulating effect. And we do it in 384 well plate, which is kind of high throughput. So if you want to assess cardiotoxicity of your compounds, or if you want to do phenotypic screens, you can use our assays. Thank you.
Thank you very much for the answer, Panida. That concludes our webinar today. On behalf of Eurofins Discovery, we would like to thank you for joining us today. Please submit your inquiries and questions to the email provided at the end of the presentation. Thank you again for your time and interest. We look forward to hearing from you soon. Thank you very much.